The overall goal of this procedure is to transfect HeLa cells with capped and uncapped mRNA transcripts of Gaussia Luciferase and assay the cell culture for luciferase activity. This is accomplished by first using T7 high-yield RNA synthesis kit to synthesize both uncapped and capped mRNA transcripts of Gaussia luciferase by in vitro transcription. Two methods of capping the mRNA are demonstrated. One uses a cap analog during transcription, and the other uses the Vaccinia capping enzyme for post-transcriptional capping. The next step is to transfect the capped and uncapped mRNA transcripts into HeLa cells. Ultimately, luciferase activity resulting from uncapped or capped mRNA is compared in the HeLa cell culture supernatant.
The main advantage of this method over setting up your own transcription reaction is that the T7 high-yield RNA synthesis kit has been tested and optimized to produce high yield and, more importantly, high quality full-length RNA transcripts. That is extremely important for successful RNA translation and expression of functionally active protein.
Though this method provides insight into RNA transfection, in vitro transcribed RNAs can also be used for other applications, such as microinjection, microarray analysis, hybridization-based blots, and RNA structure and function studies.
The DNA template for this example reaction is a linearized plasmid containing the Gaussia Luciferase or GLuc gene with a T7 promoter. Both capped and uncapped products will be synthesized. Begin by thawing the components from the T7 high-yield RNA synthesis kit and keep them on ice. To avoid RNase contamination, wear gloves and clean the bench surface, pipettes, and other materials thoroughly.